Great. So, thank you to the organizers for having me. The reason maybe I squinted at the idea that you're the first is, in some ways, the agricultural researchers are way ahead of us on a lot of this in terms of looking for genes or at least lineages and strains that control the variance of the livestock. So yes, I'm delighted to come to you and talk about two recent studies. So I don't know if anyone read the abstract, but I will talk about that work in the second half, but I added slides to talk about a very recent study we did that's not published yet, but the results are very exciting and I think very relevant to the group. To introduce the philosophy of our lab and our practice, we are interested primarily in behavior, but when you dig into the details of behavior, you find individuality is a tremendous source of the sort of spectrum that you see in behavior. And we found this when we were trying to do traditional, QTL type mapping for genetic control of species differences in behavior, but then discovered because we had an assay that measured individual behaviors, but the individuals are all different within the isogenic strains that we were trying to map the difference of. And we said, you know what, this is more interesting. Let's just do an enormous pivot and make this the mission of the lab going forward. We combine imaging of behavior. So this image on the left represents a fly, and it's walking up the stick, and it comes to this branch, and it's got to decide which of the left or the right branches to continue walking on. And this is is a representation, a very pretty photographic representation of the behavior I'm going to show you most of today, which is has become one of the focuses of our lab for the simple reason that we can measure this behavior in very high throughput. And for lots of the questions that this group is interested in, like what is the distribution of behavior across individuals? What are the distributions of higher order statistics like variance across genotypes or across perturbations of neural circuits? Let's try to use the Drosophila genetic toolkit to sort of explore this at screening scale. If you do this, you have to kind of commit to a behavior that you can measure in very high throughput. So we've gone some distance in this weird behavior of flies turning left and right by virtue of the fact that it's quite easy to measure. The central image represents computational modeling. This is the output of a simulation of the antenna lobe. So we're also interested in simulating brain circuits to see if we can explain mechanisms of individuality that way, so in silico brains. There's a whole bonus talk, somehow if I finish 20 minutes early, I'll show you that one. And then we poke at circuits using that transgenic toolkit also. So that's the central complex in flies, which is the navigation center, where the animals maintain a compass representation. So they will pick a landmark like the sun, and then use that for their navigation, and if they turn to the left 45 degrees, the representation of the position of the sun will turn by 45 degrees, so they can maintain a bearing. And that obviously, you can imagine, is going to be relevant to how the animals generate a set of statistics in their choices of going left or right. It's what are they using as bearings to choose their turning behaviors. Okay, this is our working hypothesis. I think this is broadly concordant with how the whole group thinks about things, that there are fluctuations at the microscale which propagate causally up to the higher level where we can observe things easily like behavior. So in the image, you can see that there's sort of gray shaded elements which represent different contingencies in the outcomes you would see at each of those scales. So down at the level of water molecules, you can see that sometimes the molecules might have a different bond angle or a different sort of vibrational energy, and that's sort of a level of fluctuations which might impact transcriptional outcomes, right? Does the polymerase fall off early? Do you get 2 transcripts or do you get 3? That's going to depend on how those water molecules are colliding into the polymerase. And then, of course, the variation in transcription is going to drive variation in the expression of cell surface markers, which may alter filopodia on growth cones, which alters how the axons find their partners and produce a circuit, and that circuit coordinates behavior that we can see at the macro scale. So that's our working framework, our sort of working hypothesis. And I think to the very first talk, this idea that stochasticity is in some sense an expression of our ignorance, this resonates with me. because you might look at a behavioral outcome and say, oh, it's really daunting. I might as well model that as a random factor. But then it turns out if you get lucky, you can find factors at these lower levels which predict the behaviors that you're seeing at the higher level. And so in some sense, you've pushed back the frontier of what you prefer to model as a random factor. So I'm happy, I like that idea of stochasticity as a measure of our ignorance. And I think I ripped off walls a little bit with this idea of like a veil of stochasticity that we can push back through our research. Okay, and we're also interested in the fitness consequences of variability. Here you can see scenarios where the fitness consequence is negative. So in the top, if you come from the tail of the phenotypic distribution, maybe that's associated with disease. And so if there's a change to the system or an allele or something that increases the variability, you could end up with more prevalence, more individuals in the tail with the disease. On the flip side of that, there are certain evolutionary fitness landscapes for which increasing the variability increases survival because it puts more individuals into some weird corner of higher fitness. If we're making a list of ways in which, sort of for first principles, we think that noise is adaptive, I would put that on the list. And there's bet-hedging, which was mentioned early on, but hasn't come back yet. We will talk about it. And then there's also like Nash equilibria. So from game theory, if you're playing rock, paper, scissors with me, the only strategy that you can play that you can unilaterally guarantee I won't be able to exploit is the random strategy. If you play any kind of deterministic strategy, I could potentially learn it and then beat you in the long run. But if you play randomly, I cannot beat you in the long run. So I think we could come up with a very nice list of probably 10 ways in which we think noise is adaptive in first principles. Okay, so let's jump in and talk about locomotor handedness. So you can see here a fly walking around this Y-shaped maze. It's one animal per chamber, which makes it trivial from a computer vision perspective to track that animal. So here there's some segmentation. It's very pixely because we don't just like track one fly with the camera, we track 100 flies with the camera. So they each get a little small patch of the image, but this is where we get that from. And then you get sequences that look like this. This is real data from 2 different flies from a 2-hour experiment. And blue hash mark indicates a choice to the left, and a red hash mark indicates a choice to the right. So you have the top fly, which has this very strong bias to making left turns, and the bottom fly with a very strong bias to making right turns. Flies will make hundreds of choices in 2 hours. So to the point about technical error, it's small in this case because you get a very precise estimate of the bias that the fly showed in the period of that experiment. And I'm going to sort of dig back into the annals of history to the 2015 paper where we first developed this to introduce some basics to you that are going to be revisited in the new work. And these are like fundamental properties of locomotor handedness in flies, one of which is it is over-dispersed. So you see a broad distribution of turning bias scores. Turning bias is just the fractional turns that are to the right, let's say. So that measure is broadly distributed. It's wide unimodal distribution. That's the solid lines here in this plot. And it's a wide distribution compared to the distribution you'd expect under sampling error. Right? So even if we assume that all the flies have identical probabilities of going left and right, if we do the empirical measurement, we're still going to see some fluctuations, and those are modeled by the dashed distributions there. So the empirical distribution is much broader than we expect under the null model where there's no individuality. It's a persistent behavioral bias. So flies that are lefties today are lefties tomorrow. And you can see that this is a pretty strong correlation. Second part of the talk, and that's what the promised abstract was, it examines the temporal dynamics of that correlation. So we will talk about that more. And as set up by Louisa, we mostly work within a genotype and within lab strains that, as long as you are complacent in how you culture them, tend to go to a high degree of isogeny. And sometimes we deliberately put them into a high degree of isogeny And then we find, not surprisingly, that the individual phenotypes they have are not heritable, right? So if you make two left-biased parents, they do not produce left-biased progeny. That regresses fully to the mean of a 0.5 turning bias. Okay, so in this first set of studies we did on locomotor handedness, one set of experiments was interested in whether or not we could identify neural circuit mechanisms controlling the extent of variability, right? So is variability as a trait under neural control? We've seen many examples of how it's under genetic control, but it is also under control of the nervous system. And this was, there's still some like rudimentary fly genetics on display here. These were mutants that were named, which were not mapped at the time. So, you dump a bunch of mutagen on the flies and you keep the interesting ones. And unless you do the hard work of mapping the genes, I mean, now you just sequence and you find it that way. But back in the day, this was onerous. And so a lot of mutants just accumulated that had mutants, they had names, but they didn't have any maps associated with them. And there's this nice collection put together by Martin Heisenberg in his lab of mutants that disrupt this structure called the central complex. And they noticed that animals with these disruptions had erroneous turning statistics in their walking. Right, so we had this phenotype of individuality in left-right choices, and when we thought, maybe the genes that disrupt the circuits that control the statistics of left-right turning will affect the variability of this individual measure of behavior. So we looked at a sample of these and found that compared to genetic controls in gray, there were 3 of these where there was a significantly higher variability to handedness in the mutant compared to the matched control. And you can see one of them highlighted there with the strongest effect, this thing called central body defect. And we dug more into central body defect, or at least into the structure that this pointed us to. This part of the central complex, this compass maintaining neural circuit. And here are some transgenic experiments where we could, using temperature, so we can convert the neurons in that part of the central complex into thermosensory neurons, where shifting the temperature activates the neurons or silences the neurons. And so the experiments you can see here in orange, this is when we have either activated the neuron or silenced it using these thermogenetic transgenic tools. And what you can see is higher variability. Right, so this is another disruption of that circuit. We have the developmental genetic disruption of those mutants, and now we have this acute post-developmental disruption of neural activity in the circuit through these thermogenetic variations. And if you sort of dig into the transgenic lines that drive expression in the circuit, we can do some intersectional logic of saying like, oh, I got a big phenotype in this line, not so much this line, and here's where those lines are expressed. And you can get this prediction that within the sort of red region marked by central body defect there, the structure that matters most for the control of handedness variability is this thing called a nodule. And at the time, we didn't know what this did. This was sort of like the first functional behavior paper about the nodule. But now we know that they contain signals of the animal's translational velocity through the world. Right, so the nodule, there's 2 of them, there's a left and a right one, and they encode activity in the nodule encodes these 45-degree projected vectors of the velocity of the animal. So old observations pointing to the nodule as controlling variability and turn bias. The green box says, just remember nodule, because you'll see it again. Okay, there was a sister paper to the neuro paper where we looked at genetic control of variability, and I think this is the paper that we were talking about, where we asked, do we see variation in variability across different inbred Drosophila lines? We used the Drosophila genome reference panel, which is about 200 lines caught from wild flies in North Carolina, and then aggressively inbred for 20 generations to produce 200 different lines, each of which has very little genetic variation within the line, but there's substantial genetic variation between the lines. And so what we're looking for is an example of like strain A has flies that all walk with similar handedness to each other. So you can see they're all kind of pointed more or less in the same direction. In strain B, there's more variation to their handedness, and usually point in all kinds of directions. So that'd be a high variability line or the low variability line. So we did the experiment of measuring variability for turn bias in all of these DGRP lines. Each dot here is one of these genotypes, and the y-axis is our measure of variability, which is the MAD, which somebody mentioned that earlier on, which I appreciate, because it's not used very much. And if we kind of highlight the genotypes that come from the tails of this distribution, this is a low variability one. And so you can see the histogram of turn biases from this line, and the little cloud of data points. Each point here is an individual flag. And then at the other end of the spectrum, you can see this broad distribution of a high variability line. So this is like pretty dramatic difference. The standard deviation is a factor of 2, and this is in a measure of behavior that goes from 0 to 1. So there's certain, it's kind of limited in how far you can push the phenotype out. Okay, and the very nice thing about getting a phenotype for each DGRP line is you can just take those phenotype scores and upload them and produce a GWAS, because all these lines have been sequenced. It's a very underpowered GWAS, so you get, it's not really like a Manhattan plot, it's like a Cleveland plot or Washington, DC, like all the buildings are at most 5 stories high. But there are some SNPs in the DGRP that pass some kind of significance threshold. And one of these is a gene called tenurin A, which is a cell surface marker that's involved in synaptic partner matching. So it mediates rules, for example, in the antenna lobe of 2 neurons are more likely to make a synapse if they express similar levels of tenurin. So pre and post-synaptic partner, each expressing high levels of tenurin, are more likely to form the synapse in medium with medium level. So this was exciting. This is, there's a nice circuit story to tell there. And as we're doing this study, this paper came out, where they mapped that ancient CBD mutant, and it turned out to be tenurin. So we had independently come to evidence of tenurin mattering without knowing it, which is really lovely. I mean, you don't worry about bias having produced that outcome, which is quite nice. Okay, so we dug into tenurin now, we were a bit more confident. So here, these are the variabilities associated with the SNPs that are present in tenurin in the DGRP. You can see the high variability and low variability SNP. Here, these are EMS, like null mutations for CBD, producing high or low variability depending on whether they're heterozygous or homozygous. And down at the bottom is a deficiency, so this is taking out a chunk of the chromosome that includes tenurin. And we also, again, went to that transgenic toolkit of flies and found an RNAi line against tenurin and expressed it panorally, I think, either in all cells or all neurons, but then did a critical window analysis where we disinhibited the expression of that RNAi line with temperature shifts in windows that we slid across development. So if you look at the top, you'll see about 10 rows. Each of these is a different treatment, and the orange region says at what point in development did we raise the temperature, which took off one of the transgenic blocks, allowing the expression of the RNAi, which knocked down tenurin for up to 3 days at that point in development. And then we let all those animals develop and close and measured their variability post-development. And that's what's plotted at the bottom part of the plot on the right, is the variability in those post-developmental flies as a function of when we knocked down tenurin. And you can see this evidence for a critical window here, where if you knock down tenurin during pupation, when the adult brain is wiring, you get this increase in the variability of the turn bias of adults. Okay, so I want to, so that's the old work. And I want to put this in the context of what I think is this really useful framework for providing evidence for the adaptive value of variable traits that Simon's put forward in this 2011 paper with sort of 6 tiers of evidence. And so he says that if we want to make a convincing argument that a system, like an organism, is doing bet-hedging as an adaptive strategy, we should sort of, work towards these increasingly stringent standards of evidence. So we can start, it's easy, identify a trait that varies across individuals. So we've done this, and I'll check that off. Identify relevant environmental variables determining fitness. And this is where I have to concede that we don't really have a theory as to why going left versus going right versus going straight is going to impact the fitness of a fly in the lab. We have some ideas. It could be like during courtship, the males follow females and sing to them to get them to stop running so that they can copulate. And if the female is trying to, has a tendency to turn left, but I'm a male, and my tendency is to turn right, I may be less effective at following her. And we did an experiment that even sort of showed a hint of this, that concordant flies, where the virgin female and male had the same handedness bias. When they were in an arena together, they spent more time close to each other than when the handedness biases were discordant. We didn't see any effect on fecundity, so it warrants follow-up. But anyway, so I'll just concede we don't have a fitness hypothesis for handedness. For other behaviors we study in the lab, we do, particularly thermal preference. We've done a lot of work on thermal preference, and temperature experience drives life history differences, and we think that drives fitness differences in place. So, Simon's number 3 is demonstrate genotype level differences in variability. You can tell that that is going to be checked already. 4 is demonstrate environment dependent fitness. So take those genotypes and put them in that environment where there's that variation that drives variation in fitness and show it. Demonstrate in number 5 here that the genotypic fitness depends on fluctuations in the environment, that you don't see it in static environments. And then demonstrate a quantitative match between the fluctuations in the environment and fitness. So I think it's very rare, especially looking at animals, that we check all 6 boxes. In fact, I can't think of any examples. I think the plant people and the micro people are way ahead of us, and you may very well look at this list and say, oh yeah, I know an example from yeast where all these boxes are checked. I'd love to know if you can point me in that direction. But I would say the experiments that I showed you satisfy 1 and 3, and Simon's didn't talk about neural circuit control, but I'd say maybe you do a 3B, which is demonstrate instead of a genetic mechanism controlling variability, a circuit mechanism controlling variability. And now I'm going to pivot to is the new story, where we looked at evidence for this Simon's category 4 evidence, which is demonstrating environment-dependent fitness. And we did this with artificial selection for increased variability. More of a proof of principle. We're not putting the animals in a natural environment where they get a change of fitness. We're subjecting them to an environment where their variability matters for their fitness. And this is work done by grad student Shraddha Lal with help from Noah, Chris, and David. There's a sort of part 0 paper, which is out very soon in G3, and the rest of it is not yet written. So this is really like new stuff. Okay, so the part 0 is Shraddha was excited about this idea of doing artificial selection for variability in a behavior, and we knew that this would be a lot of work. And this was also like middle of 2020 when we didn't have lab access. So we said, okay, let's do some work which might give us the best chance of future selection experiments to work with. And let's model potential different strategies of implementing this selection. And the 2 forms of artificial selection that we entertained, one is mass selection. So here you can imagine your population of organisms, in this case flies, and the darkness of the dot indicates their phenotype. So we could take this population, measure them all as individuals, and then keep a sample from that population which has high variance, right? So if you notice the dots that get kept at this point, there's big differences in how dark they are. So that's keeping a high variance sub-distribution of those animals to get the next generation propagated, and then eventually, presumably after many generations, we get a permanent change to the variability. The alternative is a form of group selection called family selection, where we'd split that population up into a number of families, measure the variability within the family, and then propagate the families that had higher variability. Right? And the reason that we thought intuitively this might be the way to do it is that variance is a trait you measure in more than one individual. So let's propagate more than one individual as the unit of selection, and that might increase the efficiency of the selection. So we did a computational model with like reasonable assumptions based on effect size distributions from the Arrows paper and 500 sites, and we thought it was reasonable. And very clearly, under a whole variety of configurations, the family selection turned out to be the more efficient predictive way of doing this. And interestingly, if you do selection for the mean, the advantage of family selection goes away. So it seems specific to selecting for variance, which seems concordant with this idea that it's about a trait that you can only measure by looking at more than one individual. Okay, so that's what we did this. We went with family selection. The experiment consisted of taking those DGRP lines, in this case 30 or 40 of them, doing a round-robin cross between them, and then allowing those hybrids of the DGRP lines to mate with each other in a cage for a random way for about 20 generations to produce a synthetic outbreak population, which we split into 3 replicates, and then split those replicates into control and selected groups, that's down here, and subjected them to 21 generations of selection, where at each generation of selection, we took half sibling flies, measured their variability, and then if they were the top variability families, propagated them to the next generation. And then did a bunch of phenotyping post-selection, which you can ask me about. Okay, and we realized in putting this talk together, we didn't actually get like that picture of Shraddha like surrounded by cages and vials, and we kind of missed out. So I don't know. Here's a picture of a tray of flies, and that's very standard fly stuff. There are funny little numbers written on the plugs, which indicates sort of the complexity of tracking which vial corresponds to which family and which ones do we keep. And then this is also Shraddha, and the picture is taken through this fly handling robot that we made, thinking a robot might be really useful for helping to do this crazy experiment. We did not do it in the end. We didn't quite get there technically, but the next time we do this, maybe the robot will help. And then these are the behavioral assays. Okay, so 120,000 flies of individual behavior measurement later. This is what the selection looked like across the 3 replicate blocks. You see in the sort of brighter color the response of the selected group, and in the darker color the control group. And so you can see, like block 2 was stubborn. It didn't really respond until about generation 15, and then we see this divergence between the selected and control groups, but blocks 1 and 3 were cooperative and diverged even back to standard generation 5. Genomic analysis is coming, like we got the sequencing data like 2 weeks ago. So I won't have much to say there, but here we go. We, as part of that phenotyping of the selected flies, looked at brain morphology. And in order to do this kind of in an objective and relatively high throughput way, we trained some neural network classifiers to take confocal imaging volumes of the fly brain and first identify the midline so that we could know left neuropil. So a neuropil in a fly is a region where you get the axons and the dendrites and the synapses. And the cell bodies kind of live on the outside, and just the information happens in the neuropils. So neuropils are like the important brain regions of the fly. And we wanted to measure them. How big are they? Is there an asymmetry in the neuropils so that a left biased fly has a bigger whatever neuropil on the left side versus the right? We did not see any of those directional correlates in the end. I'll just spoil that. Okay, so we have this nice tool for doing segmentation, and the first experiment we did was to compare the selected and control flies, so selected flies, control flies, dissect, stain, and image them, do this neural network segmentation, and then compare the segmentations to see if there are differences that align to selection versus control. And indeed there are, and it's the nodulae. It's those structures that we found in that mutant and then circuit thermogenetic manipulation experiment back 10 years ago at this point. So one of the effects on the brain of being in a selected group in this experiment was smaller brain overall. And so you can see that here in the measured volume in selected versus control flies in the central body, that's the whole central complex. The ellipsoid body, fan-shaped body, and the protocerebral bridge are these individual elements. They all got smaller because the brains overall got smaller. But the nodulae got bigger. It's like the one neuro pill that bucks the trend. And to kind of, oh yeah, this is a really nice visualization that folks put together. So here are the brains we dissected from the control flies and the selected flies, placed basically into a histogram. So it's like a horizontal histogram that goes to the right with the actual images you can see. And I think you can see by eye that the sort of sage-colored and yellow-colored blobs on the right are indeed bigger. You can just see it by eye. Okay, the other corroborating piece of evidence that came very recently examines isogenic flies, so not the selected and control flies, so just basically pick a random transgenic genotype from the lab, measure the turn bias of individuals, then take them, dissect, stain, and image their brains, do the automatic segmentation, and ask, do we see correlations between the measured neuropil volumes and the measurements of behavior that we had paired for those individuals? And this is what we saw in this analysis. There's a lot of significant covariation in these brain structure sizes. A bigger brain of a fly, that has a bigger head, is going to have bigger individual neurons. But the structure that stands out as having the strongest correlation between the volume of the neuropil and the magnitude of turn bias is once again these nodulae. And you can do sort of a Bayesian analysis where you model out the covariance, and then the nodulae are the only significant neuropil that correlates with the magnitude of handedness. So takeaways from this part of the talk. Flies have persistent, non-heritable, idiosyncratic behaviors. I imagine everybody is on board with this already. Neural circuits control the extent of behavioral variability. This raises the possibility that the brain is like dynamically tuning the degree of idiosyncrasy, but maybe in response to stress, it can increase its variability. This would be amazing, and we've looked for it, but not really seen it. But I think there's work to be done there. Genes control the extent of behavioral variability, and it is possible, proof of principle, to evolve the extent of behavioral variability, which we've done with artificial selection, and very pleasingly, genetic screens and circuit screens and GWAS and experimental evolution and correlating idiosyncratic traits within a genotype all provide convergent evidence for mechanisms regulating variability. Specifically for this handedness behavior, it's something to do with these nodulae that encode velocity information. Okay, that's part one. And let me see how I'm doing for time. Okay, all right. I'll go pretty quick through this part. So this was the promised part in the abstract, and it's on the question of drift in the individual behavioral phenotype as a possible bet-hedging strategy for unpredictable worlds. And this was led by a postdoc Ryan, who's just started his own lab at Colorado College. Okay, and here's the obligatory Harvard slide of the funding for this project was canceled. And if I were presenting to an American audience, this would be the moment of warning that, Harvard's getting it first, but the cuts to science are coming for everybody. So, we need to be ready to be political, but we're also probably going on hard times. And since this is an international audience, I will say, I hope you all can keep the flame of science alive, and if you can do anything to convince your institutions to make opportunities available for trainees to come and be in your labs and seek out a scientific lifetime in Europe, it'd be amazing. Okay. All right, so back to some old Buchanan et al. 2015 data. You've seen this already. Here's flies and Y mazes, and that day-to-day correlation that shows the day-to-day persistence of handedness. If you increase the interval between those tests, so instead of testing day 1, day 2, it's day 1, day 3, day 1, day 7, you get these plots. So here, this is the correlation of the bias they show on day 1 and then on day 3. And here's day 1 and here's day 28. If you're a fly, day 28 is approaching your lifespan. So this tells us that there is a consistent bias, turning bias, that flies have their whole lifetime. But I'm sure you've noticed that the correlations get weaker. So something is changing on long time scales so that the correlation in the bias they have at the beginning and then later it goes down as a function of time. And that's plotted here on the right. This is the R value of these experiments as a function of the interval between the tests. And it goes down. This is not a phenomenon that's specific to locomotor handedness. We did an experiment where we wanted to characterize the covariation structure of intergenotypic variation for behavior. We measured about 200 behavior measures for each of 300 flies while maintaining their identity. So we could make this big 200 by 200 covariance matrix. That's illustrated up there just so we can brag a little bit. But part of the supplementary data for this experiment was to subject a different batch of flies to repeated measures over 15 days, because this is a 15-day experiment, of the same assay on each of those 15 days, or 10 days, you can see on the x-axis here. And what's plotted is the R value between the measurement on day 1 and the measurement on day n as a function of n. And for something like 60 different behavior measures that are here. You can see basically all the lines go down, right? Most of them stay above 0, but they all kind of go down, and that's the same long time scale changes happening in the behavior. Okay, so we wanted to study this explicitly, and here's where Ryan's project starts. He did this experiment where he put flies in circular arenas. Flies' tendency to go clockwise or counterclockwise in the circular arena is correlated with their tendency to turn left or right in a Y-maze. So this is probably the same thing. So he had 60 flies, and these arenas had food. So the flies would stay there continuously for 4 weeks, except for the times when, because we did like rabbit females, they would lay larvae in the food, and then the larvae start disrupting the food. Sorry. Okay, so getting over a cold. When we swapped out the food, with the this happens sometimes. The first time this happened to me, I was lecturing in my class, and I didn't know what was going on, and it's like a laryngeal spasm, and I like freaked out and like had to leave and get some water. Anyway, you're seeing a different version of it now. Okay, we should be good now. All right. Continuous 4 weeks of tracking, except when we swapped out the foods. So basically, like 4 weeks of tracking, and then every 3 days there's like a 2-hour gap in the recording. And it produces data that looks like this. So here is 100 hours. Each different color is a different fly. So you have a faded red line, and you have a dashed red line, and you have a solid red line. The faded red line is the 1 hour rolling time average of the clockwiseness or counterclockwise of the behavior they showed in that minute. The dashed line is the 24-hour rolling average, and the solid line is the average over the whole experiment. And what you can see is that, except for the average over the whole experiment, all of these lines are fluctuating. And this is a signal that there is power, meaning there is meaningful changes in the bias of individuals at multiple timescales of the behavior. And because we had a continuous recording, we could do physics digital signal processing analysis called a power spectrum, where we measure the amount of power as a function of timescale in that signal. And you see this kind of fat curve that extends over many timescales. So it's not quite a power law, 1/f distribution if you're familiar with that, but it's a signature of behavior that is changing on essentially every timescale you look at. We have this hope that there would be like a spike in this plot, which would say all of the energy in the timescale of changes is on a timescale that aligns maybe to the circadian rhythm, or aligns to the timescale of translational changes and like protein being overturned. We didn't see that. So it suggests that there's many independent mechanisms driving the fluctuations. Okay, so let me ask, why do you think behavior might be organized this way? To get at this idea, we're going to play a little eco-evolutionary game, and here are the rules for it. You get to produce two progeny. The selective environment is unpredictable, and I will reveal it to you on the next slide. So your choices are to raise your right hand if you think both of your progeny should have the right phenotype, and raise your left hand if you think they should both have the left phenotype. And you can raise both of your hands if you want one progeny to have each phenotype. So make up your minds and raise your hands. Okay, all right, keep them up. And here is the outcome. The environment has fluctuated, and left progeny survive, and right progeny die. So put down your right hand. And those of you who still have your hands up have gone on to, found the next generation. You have like non-zero geometric mean fitness. Good job. And it's very interesting because like normally I do this and like everyone bet-hedges. But somehow this audience of all audiences is like ready to like commit to left or right, and there's some psychology or something. Okay, so game two is you get to produce one progeny, and the selective environment is going to change across days successively. What's your strategy? This one's a bit more open. No, it's different rules. Any thoughts? Yeah, you kind of want it to change, right? Right, so this is what we thought is maybe is happening with these drift, is that it's a form of bet-hedging happening within a lifespan. And so this is what we ended up modeling. I think I'm gonna end up slightly over time, so I'll try to just go quickly through this and leave time for questions. Basically, that's the idea. Is that the reason behaviors may be changing within an individual's lifespan is that they may be implementing a form of bet-hedging against fluctuations in the environment that happen on timescales faster than the generation. And we did some formal modeling around this. We had a proof in the paper, which was very exciting. And we had several proofs. And like the simple one is illustrated on the left. This is the idea that if the environment has two states and the phenotype has two states, the degree to which like the organism should bet hedge for the next round of selection, like what fraction of its progeny or with what probability should it switch its phenotype at the next time point, corresponds to like the probability that the environment changes. So this is like a form of Kelly optimality, if you know that. So the optimal strategy is for the organism to switch with a frequency that matches the frequency of changes in the environment. The assumptions you have to make here about the strength of selection and sort of the diagonality of the survival that you can only survive if you match the environment correctly go into this proof, and you could challenge those. But we did the same sort of bevy of experiments that we've done for individuality, like asking, does the rate of drift vary across genotypes? And it does, and I'll skip the modeling, except to say we use Bayesian inference to estimate the combination of like static bet-hedging versus drift bet-hedging that the animals used, because we measured the biases of individuals over time for a variety of genotypes, and you find this two-dimensional strategy space of a combination of bet-hedging and drift, and it seems to differ by the different genotypes. It's affected by neural state, so if we manipulate serotonin either pharmacologically or with mutations like de novo CRISPR mutations in this case, we can change the drift strategy by increasing the drift rate. And then we produce this forward evolutionary model where the model keeps track of the number of individuals of a given phenotype at a certain age in their own life, at a certain time point in the simulation. And the key things in the model are that for flies that are newborn, that's this lowercase, there's a bet-hedging term, which is how much variance do they come in with at the start of the experiment. And for flies that are currently alive, there's this drift term, which says how much does their individual bias change from day to day. And we could set these parameters and run the simulation and get a prediction for the fitness effects of the joint bet-hedging drift strategy as a function of the environmental fluctuations. So here there's a particular environmental fluctuation and we can give it combinations of bet-hedging and drift and get a fitness landscape out of that. We can give it different kinds of environments. So here's an example of an environment where things are changing so fast, you might as well not be random at all. Like, you're just gonna sort of ride over all that randomness. This is like, if I was to try to change my phenotype in response to like how bright it is. But I'm like going inside and outside of buildings. I can't change my phenotype that fast. I might as well just pick a static phenotype because that fluctuation is too fast to be relevant to the changes of my phenotypes. Whereas if the environment changes slowly, maybe a bet-hedging strategy makes sense in this example. We explored the phase space of this, and the take-home messages are that strong, slow fluctuations favor bet-hedging, moderately fast fluctuations favor drift, and long generation times favor drift also. Particularly what this means is that if an organism has to survive a long time through many fluctuations before it can start giving birth to the next generation, it needs some strategy to survive those fluctuations up to the point it can start to reproduce. And then we applied this to some real-world environmental fluctuation data, and this is a fun analysis here. So every point here in these plots, these PCA plots, is a natural environmental time series, like the cloud cover at a particular neon, NOAA weather station in a particular place in the world. And these are like daily time resolved series. There's like pollen counts, and cloud cover, and humidity, and sort of anything we could throw at it. And then we ran it into the forward model and asked, at the optimal strategy in that fitness landscape, are they doing bet-hedging? Are they doing drift, and how much of each? So in this phase plane, that red region corresponds to like, don't do any random strategies. Those are environmental signals for which it's like, it's the luminance change. It doesn't make sense to have a stochastic strategy for it. And then the bottom right in the green circle are the environmental signals where maybe it makes sense to do a combination of bet-hedging and drift. And then we can ask, because these are real signals, what do they correspond to as far as like the type of environmental measure, or what climate type was the weather station that produced them? So for example, just looking at the bottom plot, oceanic environmental signals tend to have the non-random strategies be the best. That's because they're like very static. It's an oceanic signal, like, the temperature in Tristan da Cunha, like some little island in the middle of the Atlantic, like just doesn't change that much in a year. You don't need stochasticity to survive it. You just need to like finally adapt to it. Whereas the tropical and tundra, Mediterranean type climates are in this like butter zone where they drive both bet-hedging and drift type strategies. Okay, so takeaways on this part. Sorry for going so long. Idiosyncratic behaviors drift over time. Behavior drifts on many timescales. Genes and neuromodulators regulate drift. And behavioral drift may be a strategy in fluctuating environments. I think we have a good theoretical basis for that. And of course, the empirics could follow Simon's protocols for this. And that's a long quest to satisfy the stringent standards there. And in the theoretical framework, drift is favored under moderate and fast fluctuations and long development times. And some, but not all, environmental signals may select for drift in a natural setting. So, thanks for your attention.
I wonder, do you assume that this decision what the fly does is already in development, or could it also be that first time the fly has to decide between left and right?
Yeah.
They all play the strategy that they then toss an inner coin and decide, and then they keep this decision for a couple of times because what you strive once you can repeat, right? And what the difference between these flies is how courageous they are to try something else until they survive.
Yeah. I think it's probably not that, but I think that's a reasonable guess. In a study we did on individuality and odor preference, about 10% of the variance of the measurement of the individual odor preference is like which of the two odor stimuli that we gave them did they experience first. So there are these kind of like initial condition effects. It's a modest portion of the variance in that experiment. For this one, I'm less inclined to say that. partly because we have found some correlates, we think, that predict not just the magnitude of the left-right bias, but is it a left or a right bias as asymmetries in this premotor region. And I think that is probably there coming out of development. I don't know for sure. Yeah.
So in your artificial selection experiment, the genetic signal that you have seen is that is presumably based on standing genetic variation.
Yes. So we established that synthetic outbred from these TGLP lines. So that is what we're assuming is the source of our gene version.
So in the first part of your talk, you showed that there's a critical period for this. And I was wondering what is special about this period for the noduli, what's happening there?
Well, I'm going to betray that I'm not a fantastic developmental biologist by saying I don't know too much specifically about the noduli. But generally what's happening during pupation is that most of the larval brain is dissolving back into a nutrient soup and then feeding the reprojection of the neurons. Some neurons survive, like the cell body survives through pupation, and so they reproject, but there's also a lot of cell division happening. So there's new adult neurons being born. All the neurons are reprojecting, or like, let's say 95% of the brain is reprojecting, including the central complex. It's disputed whether the larva has a central complex at all. I haven't seen much that's convincing. Maybe Austin's team has opinions on that, I don't know. Sorry, jokes. Yeah, so forming the brain is really the short answer. There's also something special about the critical window with respect to tenurin A, which is a spike of expression level of tenurin A, also coincides with that window.
So my question was like, did you look at all that like morphological asymmetries that could also?
Yeah, so I skipped it because it didn't have any interesting signal.
Right.
But that's like, honestly, was the main motivation. And that's why like the initial image segmentation finds the midline so that we can do left versus right. So we looked at left-right asymmetry in the neuropil. Does it correlate to left-right asymmetry in behavior? And didn't see anything.
Like across the whole body, like for example, length.
Yeah. Hard to do. So in the initial Buchanan study, we measured the term bias and then tore off all the lengths and did length measurements, which was pretty rudimentary. And at best, we could explain 10%, but it did not survive any test for multiple comparisons kind of thing. So I don't really think there's a signal there. But also that's very rudimentary because like the mechanics are complicated. And there's like 7 joints on each leg. And so, just sort of summing up the lengths, we have the thought that like, you imagine that like if my left leg is longer, it'll push me to the right. We did some amputation experiments and it went the opposite way. So if you like shorten the left leg, you might think that would introduce a left bias, but it actually introduces a right bias because they use their tarsal claws as hooks to sort of pull themselves in the direction you want to turn. And so if you cut off the end of the hooks, they can't pull themselves as effectively. So didn't see anything. We also, in the same flies where we measured the leg lengths, dissected the abdomen and confirmed that the like gut chirality was standard for all the flies. So there's a fixed gut chirality, the same way we have, and there weren't any inverted flies.
The step length?
The what?
Step length?
Stride length? Yeah.
Step or stride length.
Yeah. Didn't do that, but could definitely look at it. So like a turn in a fly does correspond to longer strides on this.
I have a couple of others. It was really interesting talk. Thank you a lot. This Drosophila is a very new system for me, and now I'm very interested to that, especially because of the X chromosome, which is expressed as biallelic in females and monoallelic in males. And I wonder if you, in your experiments, had sex difference, and I saw that, for example, it looked like there was some bimodal distributions of the histograms, but I don't know if it's related to sex differences? Do you see any?
Well, let's look at the slides after, because I don't, in my mind, we do not see bimodal differences, we always
No,
see this.
it's just, I know, the histogram of the brains you put there, if they're, for example, on top you have the female, and on the bottom you have the males, or something like that. No.
Well, those experiments where we did the brain morphology, all females.
All things again.
But I meant to ask you about this, but your poster was too popular, so I didn't make my way to the front. Because we did expect that males, because they double the expression of the X, might have higher variance in sex chromosome expression. And so we thought maybe males would have higher variance in behavior because of that, and have not seen anything. So we did a study where we took all the Y-maze data from all the other projects and put it together, and where we had sex as an annotation, looked for differences in sex to see, like, if we throw a lot of power at it, what do we see? And the answer was like, yeah, you can measure these like less than 5% differences in variance, male to female, but they're not consistent by sex. So for some behaviors, it's males that are more, for some, it's females that are more.
And for the ones where you have the mutation, from your first part, which increases already the noise, or maybe the effect is bigger.
I think 10 is on an autosome, so I don't know that we would predict.
It's on the X chromosome show.
Did I?
Yeah.
Is it on the X?
Yeah.
We didn't, I mean, those experiments, so all the screen was females. Females have various practical advantages, but like I said, I mean, maybe, yeah. I think I can't provide a clear no. That a variant dependent difference might be there.
I'll ask you the same two questions.
Yeah.
About the variant selection experiment, that is super cool. But first, that it responds so fast, and second, that it actually works. And then I wanted to link it a little bit to what Julian was in the discussion before, of this idea of, are we controlling noise to reduce it? Is there something that actually actively enhances it?
Yeah.
And then I guess the other question, if you want to comment after the answer, is, did you relax selection and does
Yes.
variance go back? Okay.
So we did 2 generations of relaxation, and we did relaxation both maintaining a family structure, but just not under selection, and also without a family structure anymore, and it was stable in both circumstances.
In
For 2
generations.
generations. And then we did another batch, which was generation 38, so it's 15 generations later, still there.
Okay, that's fantastic, because with some of you, we were discussing this idea of whether there are actually mechanisms that keep you,
Yeah.
instead of controlling, just kind of push you out, and if this is kind of stable, what is your feeling that the mechanism could be?
I guess we have thought of this, and our little toy model kind of is consistent with this, that we're fixing some of this. And that if it's a polygenic trait, we're fixing some portion of them that'll make it permanent, but.
Second question. Also, because we have discussed this with some of you, is this idea, so I'm always amazed of how huge the space, the behavioral space that you can reach within a genotype is. But we work with other populations, right? So the question is, what's your feeling on how much adding genetic variation to this equation I don't know, expands the landscape you can explore, or actually it's not really as important as what one genotype can do?
Yeah. I'll give 2 answers, neither is probably exactly what you want. But if we start with an outbred population, measure a variance of a behavior, inbreed the population, and then measure the variance again, it tends to go up. And this is maybe counterintuitive because you'd say, I want some of that genetic variation to drive variation in the behavior. I think we interpret this as like, yes, that's happening, but what is also happening is that you're homozygosing a bunch of weird alleles, and you're stressing the animals, and so you're seeing a variance effect due to the homozygosity. And indeed, if you take like 2 inbred lines that have high variability that way and produce isogenic heterozygous hybrids from them, they tend to have lower variability. And then the other answer is from that decathlon study, where the covariance structure in the outbred lines was more or less the same as the inbred lines. And we thought that, by adding alleles that differ across individuals, we would be sort of increasing the strength of correlation, or at least the eccentricity of that covariance matrix, and didn't really see that. So maybe it works through because the mechanism has to run through neurons, or probably runs through neurons. It's like maybe the variations that are there in an isogenic setting, going through the bottleneck of neurons produces the same phenomenology as like when there's genetic variations running through that bottleneck. Maybe.
I have another question, have you tried maybe thinking about what you were saying this about the number of possible behavior patterns, complexify the decision making, give it 3 branches instead of 2 or 3?
Yeah. In a general way, yes. I thought about this actually most on some of the chemotactic behaviors, we do order A versus order B. But I've often found myself thinking maybe we'll see a much richer individuality if we give them 5 or 6 choices. Yeah. We did do it. I thought you were going to ask an experiment where I could answer about this enrichment study we did, where we grew them in diversified environments to ask would that sort of increased environmental variation drive more behavioral variation, or would the more naturalistic environment of the enrichment drive canalization, so reduce variance? There's small effects, it varied by genotype, and it varied by age. So complex.
Concerning the last part of the talk, do you think that with climate change, there will be some population that will be more prone to selective for weight or packaging, depending on the time scale on the extreme event?
So my best understanding of the predictions of climate change as respects these signals, because as best I can tell from the literature, the predictions about to what extent will climate change drive increases in the variance of the fluctuations, that's complicated. Clearly it's driving this increase of mean, but what is it doing to the variance is less clear. So I think in cases, and maybe this is regional or signal specific, where it drives an increase in variance, then it'll push the animals towards having an adaptive value from the stochastic strategies. Another study where we did do sort of an explicit climate model of adding 2 degrees to temperature fluctuations. What happens is you see a pressure to move away from bet-hedging into adaptive tracking, where there's heritable variation for the traits, so that then you can adapt to the mean. And so now at least your phenotypic distribution is centered on the sort of adaptive zone of the increased temperature. And then you can potentially do a stochastic trait at that point. But it's increase variability first, and then maybe decrease it after that.
Okay, and then let's thank Ben again.